For most accredited zoos and aquariums, a main part of their mission statement revolves around working to protect and conserve the species in their care. There are various genetic techniques that are used to aid research efforts and ensure the propagation of both captive wild species in years to come. These efforts focus on in situ conservation, in which a population is managed captively. After successful captive management, the hope is that individuals can be released into wild habitats and can be managed through ex situ conservation in their natural home setting. In this video, we will discuss how genetic techniques are used in the individual species and population level to protect and conserve threatened species.
In conservation, it can be useful to study an individual's genetic makeup in order to determine the best breeding programs for species, ensuring an increase in genetic diversity and viability of the population. Records on all captive and some wild individuals are kept for about 850 species. These records are called stud books. Shown are various covers of stud books from different species. These stud books include information about the age, sex, parentage, and location of individuals for the species. Shown here is a diagram from the International Cheetah Stud Book, which shows the age of individuals in the species and the number of males and females. Shown here is a table which displays parentage. You can see the stud number of the offspring, the sex of the offspring, its birth date, its father and mother, and where it was born and on what day. This is information for those individuals born in 2015. Next, in this chart, you can see the location of the individuals within the cheetah population. This information helps scientists to gather information about the genetics of the entire population.
Genetic techniques are useful in determining certain information required to maintain records such as sex, parentage, and pedigree. For example, many bird species are monomorphic, meaning it's hard to decipher between male and female individuals. Therefore, to determine sex, an egg membrane is removed from the shell and enzymes are used to extract the DNA. The DNA is then amplified using a polymerase chain reaction and placed in agarose gel to undergo electrophoresis. In birds, females are the heterogametic sex with a ZW set of sex chromosomes, while males have a ZZ set of sex chromosomes.
The W chromosome, seen here, is longer and therefore more impeded by the gel matrix in electrophoresis. Therefore, in this gel electrophoresis, you can see that the female has two distinct bands, the W and Z chromosome, while the male has two Z chromosomes. Using this information, we can determine whether the bird is male or female. This is just one example of how individual genetic testing can help us gather more information about how to conserve species.
Genetics is important in helping scientists understand the differences between species and subspecies and how closely related they are. For example, in the case of the northern white rhino, which is extinct in the wild, scientists are comparing the entire genomes of northern white rhinos to southern white rhinos to determine how closely their genomes are linked. To do this, they are using a vital conservation technique, DNA cryobanking. This technique, in which oocytes, cell cultures, sperm, and embryos are cryogenically frozen and stored for later use, is most popular in San Diego's Frozen Zoo, which houses almost 1,000 taxa of species. It is from such samples that the rhino genomes are being sequenced. Additionally, if the genomes are found to have a high degree of similarity, resources from the Frozen Zoo will be used to create northern white rhino embryos, which will be placed in southern white rhino surrogates.
Another aspect of determining species via genetic techniques is looking at hybridization events. One case study of this examines the European wildcat. These cats are not endangered, but some live in increasingly fragmented habitats. There is a captive population among European zoos, and some of these individuals have been released into Germany. A study was conducted that sequenced the mitochondrial maternal DNA of European wildcats from the wild populations and captive populations. It was found that 92.3% of captive individuals were hybrids of domestic cats. This is an alarming figure given that some of these individuals have been released back into the wild population. In order to conserve a species, scientists must conserve purebred lines, and therefore the captive populations of European wildcats are not viable to use for reintroduction purposes.
Genetic techniques are also vital in ensuring genetic variance among a population is maintained. This is why it's so important to maintain good records with as many animals' pedigrees as possible so that captive breeding programs can be successful. If sufficient diversity is not achieved, then inbreeding depression can occur. This is why a loss in diversity in a population leads to greater homozygosity in individuals and therefore a greater expression of harmful recessive mutations. One major risk to the genetic diversity of captive populations is genetic drift via the founder effect. Captive populations are relatively small and originate from only a small portion of the wild population. Due to this, the individuals in the population may not be an accurate representation of a random sample of genes from the wild population. This is the founder effect, in which the new population has reduced genetic variation from the original population. Over the years, this can reduce variation, which will be amplified as captive breeding occurs and inbreeding depression can be seen.
One example of how zoos are using genetic techniques to reintroduce inbreeding in captive populations is through paternity testing with gray apes. Chimpanzees and bonobos live in multi-male, multi-female groups, and it is not often possible to tell who the sire of each offspring is. In the case of gorillas, the silverback male is generally the sire, but occasionally younger males will impregnate females. Therefore, it is important for zoos to test for parentage of new offspring in order to maintain records of known parentage so that inbreeding can be avoided. For these tests, a small sample of hair is removed from the apes and the DNA is tested for microsatellites, sections of repetitive DNA in the genome. The same alleles are compared in mother, potential sires, and the offspring. The scientists can then match up allele and the child inherited from its father, and the potential sire with that same allele and parentage is determined.
Zoos often have programs in which they help animals on the endangered species list grow in numbers. As discussed, the major approach they use to do this is through breeding programs. In a way, these programs are playing God. They are taking species that have been taken out due to disasters or lack of habitat and attempt to save them and then reincorporate them into the environment. For many years, there were no captive breeding programs, and ecosystems still survived with natural transition from species to species. Oftentimes, many of the species scientists are fighting to protect would have died out long ago due to pressures on their population. These pressures could have been due to either natural or anthropogenic effects. It is debatable whether conservationists have an obligation to become involved when it is human impacts that have caused species decline in the first place, or whether the species should be left unmanaged regardless. Additionally, the conservation of some populations could potentially increase the survivorship of those that would have died out due to natural selection in the wild, and therefore may mean the captive population is less genetically adept at survival in the natural environment than their wild counterpart.
The goal of captive breeding programs is to integrate individuals back into the environment. Sometimes this can be beneficial integration, in which adding species actually helps the environment flourish again. One great example of the debate regarding the reintroduction of species is the reintroduction of wolves into Yellowstone National Park. The wolves in Yellowstone were wiped out due to hunting in the 1930s. However, in the 1990s, the wolves were reintegrated into the park. Because humans had originally wiped out the population, it could be argued that it was our ethical responsibility to restore it. This had a great impact on the ecosystems. The wolves helped reduce elk and deer population, which in turn increased riparian foliage. This helped restore and clean rivers. In turn, this allowed keystone species like beavers to return to their habitat. However, the reintroduction of wolves cost ranchers huge sums of money, and they lost considerable livestock to predators. This example articulates the importance of debating the pros and cons of reintroducing a species to an ecosystem. Revive and Restore is one program looking to reintroduce some already extinct species back into the environment. They seek to enhance biodiversity by sequencing the genome and revive extinct species from fossil DNA. Two of their projects include the revival of the woolly mammoth and passenger pigeons. It would be especially important before the reintroduction of already extinct species to consider the economic and ecological implications of introducing such species into the wild. Will the species compete with native wildlife? Will it hybridize with natives? Will the species change the ecosystem or habitat in a way that will harm or aid other species? These are all vital questions to pose before any reintroduction can occur.
Overall, the use of genetic techniques to conserve zoo animals is an important tool that can be used to generate, maintain, and protect both individuals and populations of species that otherwise might be at risk for endangerment or a loss of genetic diversity due to the conditions in captivity. Research in the field of zoo animal conservation is growing and has been shown to have greatly beneficial effects, as seen in the case of chirobanking done in the San Diego Zoo, along with the study on hybridization and the releasing of species done in regards to the European wildcat population in Germany.
This research has given scientists the opportunity to observe genetic trends and data and apply this material to their breeding, housing, rehabilitation, and release programs to strengthen the genetic integrity of these species as a whole. However, this research and the idea of tampering with the species has not been entirely well received by the community. Several claims have emerged that stand opposing the research done on genetics and the conservation of zoo species, as well as the applications it proposes. Just one example is the argument that conservation projects are playing God when they engage in activities such as removing species from a destroyed habitat and attempting to reincorporate them using genetically based means.
While ethical issues and questions over the rationality of conservation in zoo animals remain, there are various benefits of genetic research in the zoo animal field, and such information could change the outcome for many scientists facing problems, either as a result of environment or genetic change. It is up to the scientific community and those working alongside these programs to see the research is used appropriately and the caution is taken when considering the potential effects that decisions regarding the genetic future of these species could have.